Hello and a very warm welcome to our webinar today. Today we're going to talk about why oxygen matters, real-time oxygen tracking in an organ on chip. Without further ado, now I'd like to introduce our speakers today. First up, we'll have Valentin Wegner. He holds a Master of Science in Molecular Medicine from the Friedrich-Schiller University in Jena. And he recently completed his doctoral work in molecular biomedicine at the University of Hospital of Jena, with his defense pending, hopefully for this year. So we're all really excited for that. Because since December 2025, Valentin is now with us at Dynamic42, and he contributes to the R&D team, specifically with his work on our gut models and the sensor technology. After Valentin, we will have Stephanie Fuchs from PyroScience, which are our partners in the development of the oxygen sensor chips. Stephanie is passionate about sensing microfluidics, especially microphysiological systems. Sorry about that. She found her passion for the field during her studies in medical physics while working on a heart-on-chip model in the group of Peter Luske. After finishing her studies, she continued with her PhD in Thorsten Meyer's group at TU Graz, focusing on the development and integration of optical sensors in several organ-on-chip systems to facilitate real-time and multiparametric monitoring. Now she supports customers in integrating oxygen and pH sensors in their microfluidic systems as an application scientist for PyroScience, drawing from her long-standing experience with microfluidics in academic research. So with that, I'd like to hand over to Valentin.
Hello, also a warm welcome from my side. Before we dive into the topic, I just want to give you a brief outline what we're going to talk about today. So I'll start to introduce us, Dynamic42, our organ-on-chip model, and then we directly dive into the main goal today, oxygen and oxygen measurement within our systems. Then I hand over to Stephanie. She'll also introduce PyroScience as a company and then talk about the technical background of our measurement. And then we wrap everything up with some examples from our lab and how you integrate that into your organ-on-chip systems. So who are we as Dynamic42? We are a spin-off company of the University Hospital here in Jena. We are placed in the middle of Thuringia in Germany. And we are surrounded by a huge biotech campus and external institutes, the university, and of course also the university hospital around. So what do we do? First of all, we offer the products and applications for you. So we offer for you the chip platform and all the consumables you might need to develop your own organ-on-chip system. But we also offer our experience in establishing new models and are, of course, sharing the models we already established within our labs. Second big chapter is the CRO, the contract research, where we are offering that you're just outsourcing your research to us. And last but not least, we are also offering training opportunities, either here on site in Jena or also on training opportunities, on workshops externally, wherever there's space and time for that. So, but what is really our main product here? We have developed the Dynamic Organ System and Developer Kit, and this basically includes everything you need for organ-on-chip. The base of that are our quite diverse biochips by now. We have single barrier model chips, double barrier models for spheroids and organoids for different amounts of cells, depending how much cells you really have as a source for your experiments. And if you need that, we can also provide pumping systems if you don't have any yet in your lab. So, and this is basically our standard chip format. It's the size of like a microscopic slide. It is divided into two chambers, like on the left and right side. So you can do two separate conditions in one biochip. And we have ports on each side to perfuse the channels. And the channels you can better see here. So we have, for the normal classic one barrier chip, a porous membrane in the middle. We can put cells on top and on the bottom, and we can perfuse both channels then separately of each other. And this we simply do by clicking our tubing, our reservoirs into a pump. We mainly use peristaltic pumps, but syringe pumps work as well. If you have already pump systems present in your lab, they are normally comparable or also usable, because we are using the standard Lewis size format for our tubings and also our ports. So, but the models itself are not the only thing really we have established in our labs. We really optimized a lot of readout options for our chip systems. That starts with normal supernatant sampling. So basically everything you can think of you would do in 2D cell culture from your cell supernatant, you can also do in our biochips, like measuring LDH or cytokines through our supernatant we have. You can also recover the tissue by cutting out the membrane out of the chip, then stain for immunofluorescence, recover the cells for flow cytometry, lyse the cells for Western blot or RNA sequencing. Also here, whatever you can think of what works in your 2D cell culture also works in our chip. But you don't have to necessarily cut the membrane out. It also works to, for example, do live cell imaging within our chip, do also functional assays, perfuse immune cells. There are quite a bunch of options there as well. And last but not least, we also established some assays for measurement of enzyme activity or also transporter function within our systems. So, but now we want to talk today about oxygen. As you know, in our body, oxygen is quite important, and we have, depending on the organ, quite a difference in oxygen levels within our bodies. That starts really by anoxic conditions in, for example, tumor tissue. Intestinal tissue also has quite low oxygen values. And on the other hand, lung models or lungs in the body, they have, of course, quite a lot of oxygen because there the oxygen enters the body. The problem is in vitro cell culture, oxygen is often neglected, or you simply place your whole plate, dish, whatever you use in a hypoxic incubator. But you can't really be sure how much oxygen is in your media, on your cells, because so far there was no convenient method available, also commercially available, to really monitor oxygen levels. And we had the same problem with the chip systems we established in our labs as well. So we have a wide range of experience with different models, but the ones we have the most experience with and really have established in our labs are liver, intestine, lung, cancer, and vasculature. And as you saw on the previous slide, they all have quite different oxygen levels. And we so far were not able to really monitor them because there was no really an application available to buy to really monitor that. That was the reason that we started to develop the DynamicOrgan O2-Sensor Kit, which is the base of the talk today. So from the consumables and the chips, it's mostly comparable to our normal kit we sell for the normal organ models. The differences are the chips, I'll go into detail in a second, and the fire plate as really the oxygen meter we are measuring the oxygen levels with, which was developed by PyroScience, and there Stephanie is going into detail. So to look at our sensor chips, the main difference is you see that here in small, in green, the dots we have in our chip integrated. I have a picture where it's a bit magnified. These are the sensor spots we are using to monitor oxygen levels within our chip. And how this is all being done, now Stephanie will tell you in more detail.
Thank you, Valentin. I will also first very quickly introduce PyroScience. So PyroScience is a medium-sized company based in Germany and Austria, and we are providing optical sensors for different applications in life sciences mainly. So also organ on chip and microfluidics fits perfectly into that. And in general, PyroScience provides three main analytes or sensors for three main analytes. The first one is oxygen, what we are also talking about today. The oxygen sensors measure the partial pressure of oxygen either in a liquid or in a gas. So you could also measure on the air side of the lung on chip, for example, but also in the medium. Then the second analyte is pH. This is only measured in water, and we have 4 different versions of that, each spanning 2 pH units, but very, very sensitive in that region. And the last one is temperature, which we also have for measurements in water and gas. So how do the sensors work? For oxygen sensors, and also actually in general, all of our sensors work with a luminescent lifetime measurement. So we have our sensitive oxygen indicator dye, and which is just get the laser pointer, is emitted, it is excited with red light, and then it emits in the near infrared. So this red light is already a good thing, so it does not interfere with the cells and also with different materials which might be in your chip. And then you can see, depending on the oxygen level, the luminescence will change. So if there is a low oxygen level, the luminescent intensity is high. And if you are at higher oxygen levels, the indicator will be quenched by the oxygen, and then you get lower luminescence intensity, but also the lifetime of the luminescence will change. The good thing about this is that this measurement principle does not consume the oxygen, unlike electrochemical sensors would do, and also it is very, very fast. And due to the red light and near infrared, it does not interfere with a lot of things. Also, because we are measuring the lifetime, we are phase-modulated lifetime measurement, we are really robust against other influences of the setup like ambient light. This measurement principle works that we have a phase-modulated excitation light, so looks kind of a sinus, and then the emission will follow with a certain time shift, or phase shift as we call it. This is called delta phi. So if you ever look at the raw data of our log files, you will see this actually. If you ever wondered about that, this is the explanation. From this, we can calculate all the way back to all different oxygen units, which you will also get in the measurement. And by using this technique, we are not interfered by any blockage of the light path, which might occur to the cells, for example, or small inhomogeneities of the illumination. So it is a really robust readout. And our other sensors, pH, work very similar, so that everything is compatible to each other. Our setups in general have 2 parts, which one is the readout device, which can look very differently. Today we will focus on the fireplate, with which the chips from Dynamic42 can be read out. And then our sensing material can be also integrated in a lot of different ways. We have sensor spots, which can be sticked into systems, complete microfluidic flow-through cells, or also we can really miniaturize it even down to the tip of an optical fiber. So the smallest one is a 550 micrometer optical fiber tip, which can be inserted into organoids, for example, but now a bit more towards the sensors as they are in the system of Dynamic42. So there we have contactless measurement, so we can really miniaturize the sensors, put them directly into the system, and then they can be excited with the same light which comes from the fireplate as it would be in the example before. The good thing about this is we can really get it in situ, close to the cells, get a very fast real-time measurement, and also everything inside the chip can be sterilized. So you don't need to worry about sterility because everything in here is sterile, and the readout is just through the transparent chip material, so there is no way that there will be any ingress of a contamination due to that or any leakage. So with this, we have a really robust system in combination with what I mentioned before, the robust readout method and the no influence on the cells, no oxygen consumption. There should actually be nothing that is interfering with the cell culture at all. And also you can just put the whole thing into the incubator and forget about it, and you will have the data of the whole weekend, for example. So there's really not a lot of effort as soon as the system is set up. How do the sensors get in that system? That are our oxygen mini sensors. So in this case, we integrate them directly into half-open chips. So we get them sent by Dynamic42. We directly place our oxygen-sensitive material on these 3 positions, which Valentin already showed you. Then we send them back. They get closed, and then you can have the whole system just completely closed by Dynamic42. So you don't need to worry actually about integration at all. Afterwards, you can just use it with our fireplate. So the fireplate is this device where it was developed for 96 well plates, or 69 well plates. So it can measure 69 oxygen sensors in parallel. You typically would plug in the 96 well plate, but for Dynamic42, you can just screw another holder on top, and then you can just easily just put the chips on top. The sensors will be aligned with the channels of the fireplate, and you can get a very robust readout every time at the same position. But you can also, whenever you need to access the chips, just get them out there and measure. This whole thing is usable in the incubator and has also integrated temperature sensor for temperature compensation. So if the temperature is changing, you will notice, and also the measurement will not be affected too much by that. Also, with this readout device, there are no optical fibers needed, so you don't need to worry about them. You can just place the chips on top and everything will be fine. So how does this now look when you really use it? This is a screenshot of our Windows software, the Pyro Workbench. This comes for free with the device. You can basically access everything you need in the software. You can start the measurement and start also the logging of the measurement, which is very important. There is a calibration wizard and a settings wizard, and you get already graphs during the measurement. To go through the main setup, I will just show you a few further pictures. So first up, the settings. So you first need to tell the software which analyte you want to measure and some sensor details. You can also organize everything in groups so that you have, for example, the top channels and the bottom channels separated from each other for data analysis later on. Then you need to tell the software about the sample properties. As I said, you can measure in liquid or gas. And then also you can select the unit in which you want to measure. We offer quite a few of them. So here's an example of what you can do for water measurements. For example, you can have micromolar per liter or milligram per liter, hectopascal, microgram per liter. So there's really a lot of options for gas. There are other options which make more sense in gas measurements. And also you can set the sample interval, which can be everything from a few seconds to minutes or hours, depending on what you prefer for your measurement. Afterwards, you need to do the calibration. There are several options for that. One of the easiest one is called the manual multi-channel calibration. They just put the whole sensors on top of the fireplate and calibrate them all at once. For the upper calibration point, you can, for example, just use ambient air as a calibration standard. So you basically don't need to do anything. And you just put everything on top. You wait for a few minutes, and then you just press take value, press finish, and you're done with the calibration. So it's really easy to do that. Afterwards, you can then start a real measurement, and you already get during the measurement all these live graphs. So we have, for example, a heat map where you can then look if some chips do something else, and you might want to react to that. For example, you can also have a multi-graph where you can see all 69 sensors at once, or with the chips, it might be a few less. And then also this multi-graph where you can then also click on one of them. They are in the same positions as the wells on the well plate. And then you can just get a zoom in on this individual graph. So that is very convenient if you want to take a closer look at one of the sensors. After the measurement, you can then also do data analysis. We also offer a second software, which is the Pyro Data Inspector. That one offers you the same kind of plots as the Workbench, but also a few data processing tools. If you don't want to use another software, you can also use basically any software which you have used before, with which you are familiar. Our log files can be exported as TXT files, and with that you can just basically import in any software that is available. So very commonly used is Microsoft Excel or Python, but we also heard of GraphPad Prism and a lot of other softwares. For Python, it might be worth mentioning we have a public package which is available on PyPy. It's called the Pyro Toolbox. That one allows you to import the data in a very convenient way into Python so that you have your data frame ready and can just start to analyze the data as you're used to it. And it also has a few nice first features. If you need to change the oxygen unit, for example, that's also available in there as it is in the Pyro Data Inspector. And with that, I'm done, and I would hand back to Valentin, who wants to show you first examples of the measurement in the lab.
I work with the intestinal model, and as Jule already introduced me with, so a lot of the establishment was done on the intestinal model, and it's a one barrier model. So we have one porous membrane in the middle, and we have on top our endothelial cells, and on the bottom epithelial intestinal cells. And the nice thing is, if we connect that to a perfusion, we get with our intestinal cells a nice three-dimensional outgrowth. As I showed you before with our chips with the sensor spots, we have one in the top channel, two in the bottom channel, one on the inlet where fresh media enters the chip, and one on the outlet where you can measure how much oxygen was consumed within our chip. And how to set it up in the incubator, it's basically quite easy. You have your chip connected to your pump, and you simply place it on the mounting frame. There's only three positions where you can choose from, but there's no moving at all. It quite sits straight in there, so no problems with the measurement at all. So, and if we look now at how this all works on the fireplate, here again on our chip, you see one sensor spot in the top and two in the bottom channel. And if we zoom in to the intestinal model, we see when oxygen enters, it is consumed within the model, and especially in the lower channel, we can measure how much is consumed, and we can live monitor that with the PyroScience software Stephanie already introduced. So, and if we look at this model, we wanted to first characterize our model as we used it before. And this we normally perfuse with about 50 microliter per minute. And as you can see here with the light blue line in our graph, that's the top channel, being basically almost fully saturated. Here to the unit, oxygen air saturation in percent basically means how many percent of our oxygen we have present, so how many percent of our 21% oxygen. So, and for both, also in dark purple, the bottom channel inlet, we basically have almost a saturated oxygen level in media. And if you look then on the consumption on the pink line on the bottom, this is the outlet of our chip. There we see a gradient, basically how much oxygen was consumed. And if we then change the flow rate to about 10 microliter per minute, for example, you can see how the amount of oxygen brought into the model in the inlet side, still the dark purple line, doesn't change at all, but the gradient gets bigger. So if you look at the pink line, both again, bottom channel, you see much more oxygen is consumed. And why is this? Because the media flows slower over the cells. The cells have more time to consume the oxygen, so more oxygen in general is consumed. And now also interesting, if you look at the light blue line, now also with this reduced flow rate, the endothelial cells have enough time to consume oxygen. So also there, the oxygen levels within the top channel is reduced. So, and now what happens if we not change the flow rate, but put it to static conditions? The color of the curves stay the same. In about 30 to 60 minutes, depending on the channel, the cells already consume all the oxygen present. And as most of the organ-on-chip systems do, they do especially on the static build up a medium exchange once a day. And now we've finally measured what that means for not only the nutrition, but the oxygen levels. With only one medium exchange, you really get, after about latest an hour, not completely, but almost oxygen-free media within our chip. So that also led now to us questioning the whole setup of the static buildup with only one medium exchange every 24 hours. But of course, we didn't only want to characterize the models we are using. We also wanted to do some biology with it. And for that, we compared two models, one left untreated and the other one treated with rotenone. And what is rotenone? It's a drug which inhibits the first complex in the respiratory chain, basically hindering the mitochondrium to consume oxygen. It means cells won't consume oxygen anymore. And if you look at the level of the gradient, the consumption of oxygen after we added the rotenone, we basically see, especially if you look at the consumption in the lower channel, the pink line, that the cells are not able to consume the oxygen anymore, which means the oxygen level within the media almost stays the same. And then if we wash out the rotenone and replace it with fresh culture medium, you can see over the course of about a day, 24 hours, slowly the cells start to recover step by step and start to consuming oxygen again, re-establishing the oxygen gradient in between inlet and outlet. So this should just be like a first glimpse into the possibilities of the oxygen measurement with the intestinal model. We are currently working with our liver and lung model to also measure the oxygen levels there, and hopefully we can soon also share data regarding the other models. But now I want to summarize what we now talked about in the last half an hour about the oxygen measurement. So now with the DynamicOrgan O2-Sensor Kit, we are now finally able to measure oxygen levels in the full organ model in the top and bottom channel separately. This is a direct measurement, no oxygen consumption, but it really helps to characterize the levels of oxygen present. This all is done with the robust red flash technology Stephanie explained, and this really works so good as it does in our chip models because the sensor spots are biocompatible, they can be sterilized, and there is no need for a direct readout or a direct contact for the readout. And last but not least, it's not an endpoint measurement. Really, it is a continuous measurement in whatever time frame you want to measure your oxygen levels. And I want to finish the presentation with our distributors. So if you're coming from the United States or Canada, Ohio Lumex would be the ones responsible for you. For Switzerland, Slovakia, Slovenia, Poland, and Croatia, we have Exxon Lab as distributors. And if you have general questions about the models, about the technique, or also for the distribution in all the other parts of the world, you can always contact us as well. And with that, I would hand over back to Jule, and we will start with the Q&A section.
Thank you so much for the insights, Stephanie and Valentin. I think that was really great. And we already have quite a few questions. So I'm going to start right away with the first one, which is thank you for showing the two-channel biochip design. Given that you have integrated oxygen sensor spots on both top and bottom foils, can this system be used to monitor the real-time oxygen gradient between the tumor layer and the stromal endothelial layer in a co-culture setup? and how sensitive are these sensor spots to rapid metabolic shifts? So I think that's one for you, Valentin.
Yes, so that's a tricky question. As I understand the question, if the gradient from one channel to the other one is measurable, as we don't have like a direct measurement on the cells, but just from media to media, it's hard to measure the changes in the consumption of the cells or how much oxygen might diffuse. But we are currently testing that with our lung model, but we don't have the final values or results to that. So for that, you could, I think, best contact us. And as soon as we have analyzed our lung data, we would go or come back to you and hopefully be able to answer these questions about the gradients from one channel to the other one.
Perfect. Thanks, Valentin. And there is a question from Lisa. Could I also use the PyroSting O2 device to measure oxygen within the Dynamic42 biochips? That's probably more to you, Stephanie.
Yes, I'm not sure if we ever tested that, but in theory, the chips are the sensors are compatible with the PyroSting. It would be a matter of checking if all of them have the right signal intensity, but I don't think that there should be too much of an issue there. The thing is that you cannot read out all of the oxygen sensors, which are even on one of the chips. So there are two systems with 3 sensors each. So you could, with one PyroSting, it has maximum of 4 channels, so you can only measure 4 sensors at once. So you would need multiple PyroStings, or you can always just measure one system. And you would need to get yourself some kind of holder to access the right position. So we don't have that ready yet. However, in theory, it could be done, at least for first experiments or something. That would be possible, I would say.
Thanks. Okay, so now Vinitra has a few questions. I'm going to start with the first one, which is, what is the limit of detection for the oxygen sensors?
Well, that's easy. Just give me a minute. The question is, in which unit? So we have in general a detection limit of 1 hectopascal for the sensors, which I will just also give it in percent air saturation, I guess. What would be 0.5% air saturation in dissolved oxygen? So this is how they are specified. One thing is that the system is at least slightly oxygen permeable. So I'm not sure if you have already tested, if you already get down to that. And it's also not the range where the cells would be much heavy anymore, I would assume. But maybe, Valentin, you can comment on that.
So for us, the limit of detection, also what we discussed, Stephanie, I think the technique is much more sensitive than we needed for the biology we are testing. So our experience is with cell culture in general, you will all know it, the cell number in general might differ simply by resuspending your cells, or there's so many biological differences or changes you might encounter, basically meaning this very tiny windows we would be able to measure are normally not needed for the cell culture, because the changes in cell number or in general the biology is too big to really need such a detection limit.
Oh, thanks. Yeah, I think that answered this sufficiently. So the second part of the question is, what is the reusability of the oxygen sensor membrane part and the cultural plates? I am not entirely sure. I think this might be referring to our biochips. I'm not sure if it's referring to like using it in other cultural plates to some spots.
I mean, if it refers to the device, that one is reusable for a very long time if it's not mechanically damaged. So that, and for the sensor spots, I think that it is, again, the sensor spots are more durable than the cell culture usually. So yeah, that's maybe Valentin, you can say more on that part then also.
Yes, so also here again, I also don't think the sensor spots would be the problem, but normally in the course of an experiment, we in one or the other way sacrifice the membrane and the cells on top. So we cut out mechanically the membrane, which prevents us from reusing the biochip. But also, if you are just taking supernatants, we tested that in the beginning. It's quite hard to get all biological residues of cells out of the chip, even if you like trypsinize it or then also try to enzymatically clean the chip. So we always recommend to use a fresh biochip for each experiment.
Perfect. And another one, can the fire plate and the oxygen sensor, can they be used at 4 degree or lower temperatures?
4 degrees Celsius. Yeah, the sensors are oh, wait, let me just double check before I say anything that's wrong on tape. No, actually, most of our sensors are specified for 0 degrees Celsius to higher levels. These are not. However, I would assume that they could also be used at lower temperatures. I'm more wondering what this application would be like for the in organ on chip. So, but maybe you can contact us and we can tell you more about the lower temperatures then. Yeah.
Yeah, makes sense. Thank you. Okay, so next one from Nadia. How much time does it take on average for a perfused system to exhaust all the oxygen within small and big medium vessels media before it needs replacement? What would be a good solution for a system that would require continuous perfusion for multiple days? For example, muscle cell tissue.
So this is what I touched with the slide with switching from perfusion to static conditions, where we saw, depending on the amount of cell metabolism in the different channels. So the intestinal cells have a higher metabolic rate than our endothelial cells. So for us, it took about 30 to 60 minutes to really consume all the oxygen. As a comparison, we have a volume of about 250 microliter within one of the channels. So that means under static conditions with about 250 microliters of oxygen saturated media, it takes 30 to 60 minutes. The thing is that as soon as you have perfusion, you get rid of the problem, because you always flush in fresh media and reoxygenated media, even if you have like a circular perfusion. I hope that goes into the right direction, what you were asking, Nadia. If not, please ask again, and maybe I can comment on that again. But as soon as you start a perfusion, you should be rid of any problems with lack of oxygen in your system.
Thank you, Valentin. Okay, and then a follow-up question from Johan. Also following up on the gradient measurement, when extracting RNA for PCR, how do you prevent reoxygenation during the harvesting process from altering hypoxia-induced gene expression profiles? How does your system help in optimizing this harvesting? So I guess the main question is like, how do you prevent from oxygen getting into the system when you take samples?
Yeah, that's mainly dependent also how you introduce the hypoxic conditions. So if it's simply within a hypoxic incubator, that makes it quite difficult. because basically as soon as you open the incubator, you have a reoxygenation of the incubator. Former colleagues of mine were working with hypoxic chambers and would then really do the whole processing of the tissue and the cells within the hypoxic chamber and hypoxic conditions, from getting them out of the chip until the lysing, all done in this hypoxic chamber. It's quite a hustle and not really easy and convenient, but that is, I think, the best way to really confirm that there is no oxygen entering or coming near the cells.
Cool. Thanks, Valentin. Next one. I'm working with iron, other metal particles in liquid. Would that interfere with the signal of the oxygen mini sensors? And also, sorry if I missed that, what is the sensitivity of the spots, especially the minimum oxygen concentration? Oh, I think, yeah, we did cover that in the questions already, so forgot about that one. But yeah, how does do iron or other metal particles interfere with the oxygen sensors? So I think that's more for...
I think it depends a bit on the concentration. If that is really making your liquid very colored, that might interfere, but typically these kind of things do not interfere with the oxygen sensors, as they cannot interfere with the measurement principle, and also they typically don't block the light path enough. However, that's also something where we can share further information if you contact us afterwards, and also can tell us more about concentrations and things, then we can give you a better estimation if we think there might be something. But typically not.
Thank you. And the next one is to precision. So what's the precision of the 3 sensor types? For example, 5 to 10%. Definitely hope I can.
I mean, for the oxygen sensors, we have, I mean, the resolution, it is about 1 hectopascal. It depends, actually, the resolution depends on the oxygen level. So the sensors get more sensitive the closer you get to 0% oxygen, and a bit less sensitive at higher concentrations. But I think that's what Valentin explained before. It usually is more than you would have, or what you need with cell culture, due to the different variations in the biology itself. However, resolution is 0.1 hectopascal at 1% oxygen, and 1 hectopascal at 200 hectopascal. So we have full data sheets where you can also get this in different oxygen units, whichever you prefer to look at. That might make the answer more easy then.
Thanks. And then one from Julia. Do you plan on establishing an integrated oxygen plus pH sensor? Valentin, one from you.
So there are some thoughts on doing that, but from the timelines we have, it's not planned anytime soon to combine both of that, I have to say.
And a follow-up from Daniel. How do you know all the consumption of oxygen is from cells and not the structure?
So the structure basically meaning oxygen binding to the plastic or parts of our tubings. We tested that and compared that also with empty chips, meaning only media and no cells present. And there we basically saw like constant oxygen levels and no decrease in between like static and perfused conditions. So with that, we are quite certain that at least most of the oxygen consumption is done by the cells. And if there's some annealing or adhesive to the plastics, it's in a level that we were not really seeing it in comparison to the amount the cells were doing.
And then just a quick question to Stephanie. Would it be practical to use the sensors on other containers, such as like tissue flask, for example, to examine the oxygen?
Yes, so I mean, these particular sensor spots, not, but we have quite a huge variety of different sensors, and we have sensor spots which look basically like stickers, which you can glue in cell culture flasks on your own. So if there's a lot of space in a bigger containers, it's not needed to have the mini sensor spots as we have them in the microfluidic chips. And if you were looking for something like that, just contact us. We have a lot of options where you can choose from. So that's completely doable.
Cool. Now we've got a range of questions from Lima Lindel. The first one is the system wireless or for data transfer? Is it needed? Is a cable needed to the PC? I can answer that. It's not. But oh no, I leave it to Valentin.
You could have also answered that. Yeah, we have the need for a cable from the fireplate to the computer with the software.
Okay. And then the second one is the PC used for storing the data or in the microcontroller? Is the one in the system?
So we have it for both, like to set up the fireplate, to set up the measurement with all the calibration, what Stephanie talked about, but also for continuously saving the data so that you're not having it somewhere all combined and then it needs to end after 5 hours and everything is saved. And if, I don't know, the battery runs out of your laptop, all is gone, but we have like the continuous saving of the data throughout the whole experiment.
Perfect. And then the third one is the same optical fiber used to excite and record the emitted light.
Actually, with the fireplate, there is no optical fiber at all. The optical channels are kind of open, or not open, but closed with glass. And you can just put it on top, so there's no real fiber sticking out like with our other devices. However, for our other devices, there are optical fibers, and there is also just one which is emitting and also collecting, which is exciting, also collecting all the emitted light.
And another one to this, does the optical channel for the sensor interfere with microscope light?
I mean, there is no, with the fireplate, there is no optical fiber. There is the full device, which is basically the same size as a microwave plate, which will be beneath the chip. However, for measurements with the microscope, you can just unplug the chip, move it to the microscope, put it on top, do your microscopy without this big metal device underneath. It's like if you would have the chip on a shaker, you could also take it off, take it to the microscope, do your measurement there, or stainings, and then take it back. So I think that's rather the idea than to have the whole fireplate under the microscope.
But also to the sensor spots, so they block some of the light of the microscope if you want to image your cells inside the chip. But our membrane is quite large within the biochips we are using, so only a small part of the sensor spot of the upper channel is blocked, because the one we have in the lower channel are on the sides of the channel and not on the optical window, I would call it, of our membrane. So you really don't have any problems imaging next to the sensors, basically.
Thank you. So one from Lisa, you saw that under static conditions, oxygen concentration is decreasing within the chips. Does this mean that diffusion of oxygen from outside the system does not have an effect?
That's a really good question. This is something we are currently further investigating. In the normal setup, what we have on one side our tubing plugged in with the Lure adapter, and on the other side a reservoir with fresh media. So we basically block the way for any oxygen to diffuse into the system. If you have a site not perfused and unplugged, we see a bit of oxygen diffusion into the chip, but to such a low extent that it's not comparable to perfusion.
And another one to hypoxia. Did you perform experiments where you induced hypoxia? And if so, how exactly did you do this?
So what we did was to use a hypoxic incubator and put the whole system into the hypoxic incubator and reduce step by step the oxygen levels within our chip. And that was quite efficiently done, I would say. If I remember correctly, we ended up like lower than 10% oxygen saturation, which equals about less than 2% of oxygen within our media. But that, I would say, highly depends on your incubator you're using.
Thank you. And another one, which might need to bring up some slides again from Oliver. Please, could you show us once again the slide measurement principles lifetime measurement?
Yes, I guess I can do that. So I can show that. The question is, what is the question? I mean, I can just keep it open now for a few seconds or explain the principle again.
Maybe just quickly summarize it again. We still have a few minutes.
So the very quick summary is we have an oxygen sensitive indicator. This gets excited with red light, then it emits in the near infrared. So it's luminescent, and if there's oxygen present, this luminescence gets quenched, so it decreases. And that's decreasing both the lifetime and the intensity. And we are measuring the lifetime. And it is this, it's called phase modulated lifetime measurements. It's actually the basic principle is published, so you can look that up, how it works. However, that's what we do basically, and with a few refinements, let's call it like that, but the basic principle is this. So that is the very brief basic principle.
Perfect. And then also again to you, Stephanie. So thank you for asking all the questions. That was from Yolanda. She had quite a lot of questions. But I was wondering for a PyroScience system, not our system, if a 96 well plate requires multiple detector sets with individual optical fiber per well.
If you use the Fireplate, no, the Fireplate has really 96 detectors basically integrated in itself. So you just need this one device where you put the 96 well plates with all the sensors on top, or anything that actually matches the 96 well pattern. So the dynamic for the two chips at least partially matches, so that you can also just simply put them on top. So you don't need to actually worry about 96 optical fibers. That would be a lot. So with that system, we got rid of the optical fibers, which we had with the other devices, because 96 optical fibers is something you will not want to handle, I guess.
Okay, so there's just one more question left, and then I think it will conclude the webinar. So the last one is, for some context, I'm trying to understand if this can be applied to microbial ecology study of extremophiles.
Yes, so that's definitely one of the examples where we feel that it's really important to measure oxygen levels, to be really sure that the chip itself is oxygen free, that the bacteria or whatever you want to grow there are able to grow. And we are also able to reach these low oxygen levels, or to measure these low oxygen levels that we can double check if the anaerobic conditions are already present. But also there, I have experience with former colleagues of mine. There, normally the problem is more to have an anaerobe chamber up and running. And if that is the case, we can definitely support with measurement of the oxygen levels then within our system.
Perfect. So thank you so much, Valentin and Stephanie, for the answers. If we did miss any questions after all, we will get back to you afterwards. Otherwise, thank you for joining today. You will receive the recording of this webinar sometime next week in an email from myself. And yeah, we'll hope to see you again next time. Thank you and goodbye.
Bye.